Welcome. In this video, I'm going to discuss about the structure of a typical dicot embryo and also monocot embryo. After that, we shall see about the seed. Now, in the last video, we understood how embryo develops in case of dicot plants, right? And now in this video, we shall see the structure of dicot embryo. Now, in a typical dicot embryo, it shows mainly two parts, one embryonal axis and another one is uh, cotyledons. So there will be two cotyledons. Of course, basing on this number of cotyledons only, we classified angiosperms into two categories, that is monocotyledons and dicotyledons. In case of monocotyledons, there is only presence of one cotyledon, whereas in this case there will be presence of two cotyledons. So now let's see the structure of this dicot embryo. So in this case of this dicot embryo, right? So we find here the presence of cotyledons. So there are two cotyledons. So these two are right cotyledons, and here is the root cap. So a protective layer that covers the radicle in the embryonic stages, and later also we will see the presence of this root cap. And here is the radicle. So this portion of the embryo called radicle, also as the root tip. So the root tip is protected by this root cap. Now we have a region called hypocotyle, which means the region below the cotyledons, and here is the portion called plumule, which is called the stem tip. So here for a sake of understanding, so I shall unfold this embryo and we shall see here. I'm going to so these are the two cotyledons. So this is how you will see the cotyledons in case of the dicot. So these are two cotyledons, and here I'm going to take this as the embryonal axis. So this is embryonal axis, and this is the cotyledon. So it's the cotyledon. Now whatever that portion above this cotyledon, I call that as the epicotyle. So epi means above the cotyledon region, and this one which is present below the cotyledon is called the hypocotyle. So that is a region below the cotyledon. So epicotyle and hypocotyle, and the tip of this epicotyle is what we call it the plumule or the stem tip, and the tip of this hypocotyle is what I call it the radicle or the root tip, right? So this is we see the parts of a dicot embryo. So this is the embryonal axis which I have drawn over here, and the region above these cotyledons, which we call it epicotyle region, which terminates with the plumule, and the region below this cotyledon is called hypocotyle region, which terminates with the radicle or root tip, which will be covered by the root cap. Right, so that part is clear. Here small information on these cotyledons too. So here these cotyledons, I can call them as embryonic leaf or embryonic leaves, and they store food materials. Okay, and then so these food materials which are stored in the cotyledons will help in providing nutrition during the process of germination, right? So that's about the structure of a typical dicot embryo, right? So this diagram is important for board examination. Fine, so now let's move on to the next one, the structure of monocot embryo. So now let's see the structure of monocot embryo. And in this case, it has again same. We can consider the embryonal axis, and this one has only one cotyledon. So the other cotyledon is not present, or in fact it becomes non-functional, right? So this cotyledon, that one cotyledon which is functional, is what we call it as scutellum, right? So now let's look at the diagram of this monocot embryo, right? So this is the diagram of monocot embryo, and this is the functional cotyledon. It's called the scutellum. So this is the functional cotyledon. The other cotyledon becomes rudimentary, what I call it's non-functional, and that remains as a small structure, what we call it epiblast, right? So this is a rudimentary cotyledon, so which means a cotyledon that became non-functional. So this is a non-functional cotyledon. So of course in case of dicots, we find both the cotyledons are functional, but in this case you will find only one cotyledon functional, whereas the other cotyledon becomes non-functional. That what I call here as rudimentary cotyledons, right? Now when we see the embryonal axis of this embryo, the monocot embryo, so we find the epicotyle and hypocotyle regions, just like how we have seen in case of dicot embryo, right? So this epicotyle portion of the embryo, so this epicotyle, it consists of the apex, that's called shoot apex. So it consists of one shoot apex, and right, this also consists of the leaf primordia, right? It consists of leaf primordia. So leaf primordium means this is a part which is going to develop into leaf, okay, upon, of course, after the process of germination. So here the leaf primordia are the primitive structures which are going to develop into the leaf. So this epicotyle, it consists of right, what we call it the shoot apex. So you can see in this case this portion is the shoot apex, and also it has the leaf primordia present, okay, in the region that's called the epicotyle. So you can see here a small portion here below this is leaf primordium. So the leaf primordium is the part, okay, which is of course it's a primitive tissue which is going to develop into the leaves, okay, after the process of germination, right? Now that's about in case of epicotyle region, right? So one more point here. So this leaf primordia is enclosed in a hollow foliar structure. So this leaf primordia is enclosed in a hollow foliar structure, okay? So that hollow foliar structure is called coleoptile, So it's a protective sheath Okay, so that you will see on the region of this shoot apex. So this is the coleoptile part. Okay, so you can see here this portion is the shoot apex, and right, that has along with that leaf primordia, so which come under epicotyle region. And then, okay, so this leaf primordia is enclosed in fact in a hollow foliar structure. You can see this is a hollow foliar structure, and that's what we call it, the coleoptile present. Okay, so that's the what is about the epicotyle region. So now, when coming to the lower part of the embryonal axis, that is called hypo, okay, cotyle. Okay, so this is the region above the what I call cotyledon, and this is the region. Okay, of course I can say this is the region of embryonal axis above cotyledons, and this is the region of embryonal axis below cotyledons. Is below cotyledons. Okay, so this is the region of embryonal axis above cotyledons, and this is the region of embryonal axis below cotyledons, right? And now this portion which is below the region of cotyledons, which is called hypocotyle. So this region, it consists of right, the radicle and the root cap. So this one, it encloses one radicle, and also there is the root cap. So in this case you can see this is the region, what I call it, the root cap, and below or behind this is the radicle. Of course, already I have mentioned that the radicle is the portion of the embryo which is going to develop into root, okay, after the germination, right? So here the radicle and the root cap, and this root cap is in fact covered by an undifferentiated sheath. Okay, that, so I can say that the radicle and the root cap are enclosed by a undifferentiated sheath, and that sheath is called the coleorhiza. So we did not see about this coleoptile and coleorhiza in case of dicot embryo. So only in case of monocot embryo you will find these special structures called coleoptile, which has a protective function okay, of this okay, shoot apex, right? And at the same time the radicle and the root cap are in fact protected by this coleorhiza. So here is the region, what I call it, the coleorhiza. So coleoptile and coleorhiza. Right, so I'll draw here the embryonal axis. So this I'll take as the embryonal axis. Okay, fine. So this is how we see the structure of monocot embryo. So right, so that's about this monocot and dicot embryo. So now let's see about the seed. Right, so this seed is the final product of sexual reproduction. So this is the final product of sexual reproduction. So in case of these right organism, so that is as I'm discussing especially with the flowering plant. So in this case, as I say, the seed, of course, in other group of what I say, the plants, of course, after the sexual reproduction, the final product will be the seed. So in this case of flowering plants also, the seed is the final product of sexual reproduction. Okay, and I can say that the seed is a fertilized ovule. So this is a fertilized ovule, because I told you after the process of fertilization is completed, that ovule will transform into seed, and the ovary will become right fruit, right, and then in the case of the zygote will become embryo. So this is what we have seen, and almost all these processes will occur simultaneously. So while the ovule is forming into seed, the ovary will develop into fruit, and the zygote will develop into embryo. So when we see right after the fertilization, the final what we see is the fruit. The fruit contains the seed. So we have is the fruit, and the fruit contains seeds, and the seed contains right embryos. The seed contains those embryos, and those embryos will have structures like plumule, okay, and radicle, okay, which are primitive structures. Okay, on germination they may start give rise to the plumule will develop into the shoot system, and the radicle will develop into the root system. Right, now in the last video I told you that by the time the seed is formed, whatever the embryo that whatever that endosperm formed inside the seed, right, of course that may be completely consumed, or it may be incompletely consumed. So here depending on the presence or the amount of endosperm that is present, okay, in the seeds, we can classify, okay, the seeds into two types, okay, depending on amount of endosperm in the seeds, right. So here I classify that into right X albuminous seeds, X albuminous seeds, okay, and albuminous seeds. So we have X albuminous seeds, right. Of course, these X albuminous seeds, I can also use another word, non-albuminous seeds. X albuminous seeds and non-albuminous seeds. Okay, right. So in these seeds, the endosperm is completely consumed by the embryo. So which I told you already. So endosperm is completely consumed by the embryo. So hence there is no residual endosperm present in the seed. Whereas in case of albuminous seeds, right, so endosperm is okay still present. So I can say a small amount of let's just say a small amount, right. So endosperm, right, a small amount of endosperm, okay, is present in the seed. So then the condition, what we call it albuminous seeds. Now let's see some examples of this, right. So where I have given examples like pea, okay, groundnut, okay, beans, etc. some examples of these okay, X albuminous or non-albuminous seeds. So I can say in these words there is no residual endosperm. No residual endosperm is present. So in case of these albuminous, okay, seeds, the albuminous seeds example include wheat, maize, okay, barley, and so these albuminous seeds in which you will be seeing the presence of residual, okay, endosperm. So in this case I say a residual endosperm is present. So residual endosperm is present. Okay. So here one more example like castor, etc. Right. So that's the two types of seeds depending on the amount of okay, endosperm in the seeds. So some seeds will not have endosperm at all. Then they are called X-albuminous seeds or non-albuminous seeds. And some seeds will have residual endosperm present. It means that they, in them, what I say, the endosperm is not completely used by the developing embryo. So I can say in this case the endosperm is not completely used by the embryo, right? So for that reason we find still the presence of residual endosperm, right? So now in case of right, some seeds, what we see usually after the fertilization process, we know that the embryo sac is surrounded by, okay, nucellus. So the embryo sac is surrounded by nucellus. Okay, I hope you remember the structure of the ovule. So when we see the ovule structure, so around this nucellus, around this embryo sac, around the embryo sac, you will find the nucellus tissue, right? So all this was, I'm not drawing completely here, so just I'm recalling. So this is the nucellus tissue, and we know that this nucellus tissue is diploid, okay, which actually surrounds that embryo sac. And after fertilization, usually by the time the ovule transforms into seed, this nucellus will completely get degenerated. But okay, there is an exception in that case. Not all seeds will show the degeneration of that nucellus. So here you see in some seeds the nucellus is still present, okay, even after the formation of the seed. So even after the formation of seed, even after the formation of seed, right, we find nucellus. So I can call that as persistent nucellus. It's called persistent nucellus. So this persistent nucellus, even after the formation of seed, the nucellus is still present. So that's the persistent. So nucellus is still present after the formation of the seed, and that's what I call it, persistent nucellus. And this persistent nucellus, okay, I call it with another term called perisperm. So I will write it again here. So persistent nucellus is nothing but the perisperm. So another word, okay, for that persistent nucellus is the perisperm. So a technical word for that persistent nucellus. And you find this perisperm in case of right, seeds of plants like the black pepper, in case of black pepper, in case of beet, etc. You will find this persistent nucellus called the perisperm. So right now, let's see the structure of seed. Now, we know that the ovule is actually covered by two, what I say, integuments. Okay, so I say those two integuments of ovule, will form, okay, the seed coat, right? So they are going to form the seed coat. So there is an outer integument and inner integument. So once recall the structure of the ovule. Okay, when we see the structure of the ovule, it is covered by, okay, two integuments, where I mentioned it as bi technique, right? And this will develop, so those will develop into seed coat. So this seed coat actually has two layers, right? So there is an outer, okay, and inner layer. Now the point is that the outer integument is going to develop the outer seed coat. Okay, so outer layer of seed coat, right? And that outer layer of the seed coat is called testa, and the inner layer of seed coat is called the tegmen, right? So this outer layer of the seed coat is the outer layer of the integument. So in otherwise I will write here, so outer integument, so outer integument of ovule will develop into testa, and then inner integument of ovule will develop into tegmen, right? And so of course these two will be the tough protective layers. Okay, so this forms a tough protective layers, okay, protecting the embryo, right? Either in case of the monocots, right, or in case of dicots. And now let's see, of course the ovule, it has a micropyle. The micropyle will remain, so it is not going to close. So you will be seeing a small opening on one side. So here I'm just drawing it. So this is the outer testa region, this is inner tegmen region, and there is a small pore opening. Okay, so that is the micropyle of the ovule. So this micropyle of the ovule will not be closed. Okay, so it will remain as a small pore. Okay, small pore, right? So it remains as a small pore. So this is nothing but the remnant of micropyle. So it's a remnant of micropyle. So the ovule micropyle will not be completely closed. It still remains, okay, in the seed. When the ovule develops into seed, even then you find that small hole. So now question is that why that micropyle is still present. So the purpose of this micropyle will, of course, in fact, is that it helps in entry of oxygen and water. So this helps in, okay, entry of water and oxygen into the seed during the process of germination. So during the process of germination, it will help in the entry of water and oxygen, okay, into the seed. Now as the seed matures, so what happens is the water content is very much reduced. Okay, so in the mature seeds, water content is highly reduced. So of course, whenever we see a seed, so we don't think that it contains amount of water, but still, as we say, it has it has still the water content present. So the amount of water present in that seed is around 10 to 15%. Okay, so 10 to 15% of moisture. So this 10 to 15% of moisture means that much amount of water is still present, okay, right, in case of these mature seeds. Okay, so by mass, so that much amount of water is still present in the seed. Even after the maturation of the seed, you will find around this 10 to 15%. And then, okay, what happens is the general metabolic activity. The general metabolic activity of seed is very much reduced. So it slows down. Okay. So seed slows down, so that metabolic activity of the seed is very much reduced and it becomes very less. So we cannot say that the seed is not metabolically inactive, means completely inactive. Okay, so it looks like, but still a small amount of metabolic activity is or will be going on. So I did not say that the metabolic activity is completely stopped, right? So the metabolic activity of the seed is slowed down, so very much less, and it will with this it will enter in a state of inactivity. So this results in okay, inactivity of the seed. So it enters into a state that is inactive, and that is called the dormancy, the seed dormancy, right? So in certain seeds, there will be dormancy and they will not germinate until this dormancy period is over. So till then they remain. So I think you can check it out, like in case of the mango seed and the tomato seeds. So after you put these seeds into the soil, you observe the number of days in which they germinate. So I think you will come to know that the seeds of tomato will germinate much quicker when compared with the seeds of mango. So the seeds of mango have a longer dormancy periods, right? And that what we mean is a state of inactivity, right? So where I'm going to discuss this dormancy, okay, so later, right? The next, now when the conditions become favorable, so the conditions like adequate amount of moisture and then oxygen and in the presence of suitable, okay, temperature. So in this case, right, the seeds will germinate. So the seeds are going to germinate, okay. And now, as I said, while the ovule is transforming into seed, the ovary will develop into, okay, the fruit. And in this case, the wall of the ovary is going to become the wall of the fruit, and that is called the pericarp. So remember this word we used in reproduction in organisms, right? So this pericarp, I can say in simple, is the fruit wall, okay. So the wall of the ovary will develop into the fruit wall, right? So the next, these fruits can be classified into two types. So the fruits are classified into two types. One I call them as false fruits, and another one are called the true fruits. Okay, so false fruits and true fruits. Now here, in case of false fruits, okay, the other parts of the flower, other parts of the flower also contribute in the formation of fruit, contribute in the formation of fruit, in the formation of fruit, right? So if it is the case, then we call such fruits as right, the false fruits. Okay, so the false fruits, we can see in this case examples like the strawberry, apple, etc., right? And if only ovary develops into fruit, if only ovary develops into fruit, then the condition is called the true fruits. Okay, like we see in case of orange, tomato, pea, etc. So in them we see that ovary will develop into the fruit, and in this case the false fruits, the parts of the flower, so like example thalamus, may also contribute in the formation of fruit. So like we see in this case of strawberry and apple, so in this case the thalamus also contributes. I hope we know the thalamus. So this thalamus also contributes, okay, in the formation of fruit. So hence in that case, right, the condition becomes the false fruits. Okay, now here the fruits can also be classified, okay, so whether they are fleshy or dry. So they are fleshy fruits, okay, and we have dry fruits. Okay, so these fruits have been classified into two types, that's fleshy fruits and dry fruits. Now in case of these fleshy fruits, we see that, okay, the seed, around the seeds or between the seeds, around the seeds or, okay, around the seed or between the seeds, there is a presence of fleshy tissue, or what I call it a succulent tissue, which means it will be having water. Okay, like we see in case of mangoes, we see the pulp. Okay, so it contains water, so I can say that as a fleshy tissue or a succulent tissue, right? If that is present, then we call that fruits as fleshy fruits. And coming to the dry fruits, okay, there is absence of fleshy tissue, absence of fleshy tissue. So if that fleshy tissue, okay, tissue is not present, then the condition becomes the dry fruits. So here let's see a few examples of fleshy fruits and dry fruits. So in this case, the guava, mango, orange, etc. are all the one who have pulp, okay, and they are called fleshy fruits. And you don't find any of such fleshy tissue like we see in case of groundnut, in case of mustard, etc. So are some examples of these dry fruits. Okay, so the fruits can be false or true fruits, right? It all depends on whether the parts of the flower also contribute in formation of the fruit. If it is the case, they will become the false fruits, and only ovary develops into fruit, okay, then the condition is called true fruits. And there may be we find the fruits have pulp, okay, so of course with the water content, right? And if it is the case, they will become the fleshy fruits, and dry fruits, okay, will not be having that fleshy tissue. In that case, of course, like we see in groundnut and mustard plants, okay. Right now, there are certain situations where, okay, the development of okay, fruit takes place without fertilization, and if it is the case, the condition is called Partino. So parthenocarpy is simply the development of fruit without fertilization. So development of fruit, okay, so without fertilization. So in that case, the fruits will not be having seeds. So fruits do not contain seeds. So I can say those are the seedless fruits. Like already you might have seen in some cases, like the grapes, okay, example are the seedless grapes. Of course, nowadays there is a great demand for the fruits, okay, that don't contain the seeds, especially like we want watermelon without seeds, right? Of course, the condition, if we are able to, okay, induce that parthenocarpy, we will be able to get right, those fruits without the seeds, right? And even we can see example of banana. So banana is also produced by the parthenocarpy, and this parthenocarpy of course can be natural or it can be induced. So as mentioned, so parthenocarpy, okay, is natural, so naturally occurring process, and it can be induced. So by applying something called the PGRs, so that's plant growth regulators, or simply I call them the growth hormones you can use, okay, or the growth hormones. So these PGRs are growth hormones, like example auxins. You can use auxins, okay, you can use gibberellins, right? So by applying these auxins and gibberellins, you can, okay, develop the fruits without the seeds. So then the condition is induced. So we can induce the parthenocarpy by applying such growth hormones, or in fact in some species the parthenocarpy is a natural process, okay. And the seeds that are produced by this process are called parthenocarpic fruit. So these are parthenocarpic fruits, right? So that's about a small concept called parthenocarpy. So with this I will end today's video. So in the next video I shall discuss about, I mean, small information on seeds, and also we shall see about apomixis and apomixis and polyembryony in case of angiospermic plants, right? So thank you and have a